Okay, let's talk about some genetics and some Punnett squares right now that have to do with your fast plants that you're germinating. So genetics. Genetics is based on DNA. We've already talked about DNA. DNA is the molecule of heredity. So it determines which traits you have as an organism, right? And DNA is made up of segments of DNA that are called genes. And these genes determine which traits you have as an organism. There are different variations or versions of genes that are called alleles. This is a version of a gene. So if you have a gene for eye color, an example of an allele is blue eye color, brown eye color, hazel eye color, etc. Okay, so those are alleles. Alleles are different versions of genes. There's a term called genotype, and this means which genes are present in the organism. Which genes are there? And it's easy to remember because look at it. Genotype. What type of genes are present in your organism? Whether it's a fast plant or a human being or hippopotamus or type of mushroom, whatever. That's genotype. Phenotype is another term we use because we can't see the genes in organisms unless we look at their DNA. But what we can see is the phenotype. And this means what are the physical characteristics? Physical characteristics, phenotype, okay? The type of physical characteristics that you see. Now, when we talk about alleles or different versions of genes, we often talk about dominant traits or alleles and recessive traits or alleles. So a dominant trait is always seen. We always see it. It is always expressed because it is dominant. Recessive traits or alleles sometimes are invisible or hidden unless you see it if there are two copies present. So you'll see a recessive trait if there are two copies of that recessive trait present. Okay, so let's keep going. Now, there are different ways that we talk about genotypes. There are homozygous genotypes where you have two of a particular type of trait. Homo means the same, right? So we always represent genes so we don't have to write out whole words with letters. And so in this case, let's use P to represent a trait of some organism, P's. So if you have a homozygous genotype, you would represent that with two capital P's or two lowercase p's or little p's like this. Now, why do you have two? You have two because you inherit two genes. You inherit one gene from your mother and one gene from your father. Whether you're a fungus or you're a plant or you're a human being, you have two copies of each gene. One came from your mother, one came from your father. Okay, so you always have two. If you have a heterozygous genotype, you have two different traits. So in this case, if we're using P's, you would have capital P and lowercase p, or big P, little p. Okay? You have two different versions of that gene. That is heterozygous. Okay, we've got some terminology out of the way. Let's talk about our fast plants for a minute. So our fast plants are plants that grow fast. Duh. That's why we use them, because we grow them. They can grow very fast. In 4 days' time, you can learn all kinds of things about their phenotypes, what they look like, okay? So we're looking at two characteristics. We're looking at color of stem, and we're looking at height. All right, so let's all use the same letters here. We're going to use uppercase P for purple colored stem versus lowercase p for green stem or non-purple, okay? Now, why are we calling purple capital P? Well, this is a hint for you. You're going to find out that the purple trait is the dominant trait and that the green trait or non-purple trait is the recessive trait. You will figure this out from the numbers that you get in the crosses that you're doing. One thing I want to say about purple. Purple means purple stem, purple leaves. If you see purple anywhere on this plant, it is purple. Okay, so the whole stem does not necessarily have to be purple. Purple coloration anywhere on the plant is a purple stem. Okay, that's a good hint for you, because you're going to see some kind of wacky things where the whole stem is not purple. Height. What about height? We have tall and dwarf. And so we're going to use capital T for tall versus lowercase t for dwarf or short in height. Okay? Here's another hint. You're going to figure out that tall is dominant, because you're going to see this in the F1 generation. So you're going to have to explain this to me, why you know these things. But I'm telling you right now that it is a dominant trait. All right, let me move on to the next page. Now, you are growing 3 generations of plants. You're going to grow one of the parental generation, one batch of seeds from one of your parents, okay? So you're going to have parent 1 and parent 2. We're not going to grow, but we're going to figure out. But the most important thing about these is that they are true breeding. That means that there are no hidden recessive traits that you cannot see in the phenotype. Okay? And that is a very important piece of information. So this is what it means. It means that both of these parents have to have one of these genotypes. And we'll talk about why in just a second. Let me get them written out. And let's look at what the phenotypes are going to look like. Okay, you see that there are 2 copies of each one of the genes. Whether it's little p's, you're going to have 2 little p's. Or whether it's capital P, you're going to have 2 of them. Same thing for the T's, same thing for the T's. No hidden recessive traits. That's what true breeding means. It means that when you cross these with each other, you're going to get the exact same offspring every single time. Okay? All right. So these are the 4 possible genotypes for stem color and height for these 2 parents. They have to be this. They are not going to be the same. So you're going to figure out, you're going to see what parent 1 is, and then you have to figure out what parent 2 is. So let's look at the phenotype. So what does this mean right here? Little p. So this is green dwarf. Remember what the T's mean. Little p, big T. Big T is green, tall. Tall. Big P, little t is purple, dwarf. And big P, big T is purple, tall. Okay? So you're going to grow P1, and you're going to figure out which of these genotypes the P1 organisms have. And how are you going to do that? Because you're going to see which of these it is. Then you're going to figure out what P2 is just by knowing about the F1 generation. Okay. And I'll turn another page here. So you're growing P1s. You're also growing F1s. So what does F1 mean? That means when you put P1 and P2, the two parents together, whoop, you get the F1 generation. F stands for filial, and that just means offspring. Okay? So you are going to grow the F1 generation, and then you can see what the phenotype is. So you're going to look at the phenotype. All right, here's another hint for you. Because this is a dihybrid cross at the end of this thing, the genotype of the F1 generation has to be heterozygous for both traits. It has to be. Otherwise, you don't have a dihybrid cross. Okay? It has to be this. So what should this look like then? Should express the purple stem trait and should express the tall stem. That's what you should see. So let's see if you do. Okay? All right. And then at Carolina Biological, they have crossed F1 with another F1 to get the F2 generation. All right, now let's take a look at that. All right, if our F1 is heterozygous, both F1s are heterozygous, oops. How are we going to put all these characteristics together? All right, here's what we're going to do. We're going to draw a big square. And in fact, I'm just going to tell you, draw 16 squares. I already know that that's how many you're going to need, so let's draw them. All right, and then what we're going to do is put all the P's and T's together, all the possible combinations. All right, let's take a look at this parent right here. Remember that parents donate one of each type of gene to offspring. So you're going to have one P and one T in every gamete. A gamete is a sex cell, either eggs or sperm, okay? So you're going to put one P and one T in every gamete. And we're going to put this parent's gametes on the top row right here. All right, so let's start putting them together. Capital P, capital T. One P and one T. All right, capital P, lowercase t. Little p, capital T. You see, I keep using different terms. That's all right. Little p, little t. The circle represents a gamete or sex cell. That shows me that you know what that is. All right, well, what kinds of gametes does this parent produce? The very same ones. Capital P, capital T, capital P, little p, big T, little p. little t. Okay? All the different types of sex cells from that parent. All the different types of sex cells from that parent. And then what are we doing? We're putting them together in all the possible different offspring that can be produced by this cross. All right, so I'm going to show you the first one here. You put all the P's and T's together in the square. Okay, and it's important to look at whether these are capitals or lowercase, okay? So capital P, capital P, capital T, capital T. All right, you see how to do this, and you can watch me do it if you want to. I'm not going to talk through the rest of this, though, so I can pay attention to what I am doing. Oops. Okay, we've got all the different offspring that can be produced through the cross of these heterozygous individuals for 2 traits. All right, let me push this up just a little bit further so we can do something else. Oops, get it over here. A little bit over this way, okay? All right, now, let's take a look at the offspring that we have produced. I think we can see it all. All right, so what are we going to get? Oh, my goodness, a whole lot of different genotypes, right? But let's look at the phenotypes. The phenotypes. What are we going to see? So purple. If we have purple, we're going to have a capital P and anything, okay? And tall, we're going to have capital T and anything as that second allele. Okay? All righty. Let's find how many we have of these. 1 capital P, at least 1 capital P, and at least 1 capital T. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 9 of those. There are a lot of those. So what is that going to look like? Purple, tall. All right, what else do we have? What if we have little p, because that means the recessive trait is going to show through, and a capital T, anything. So what is that? That's green, tall. That's tall. There you go. How many of these do we see? Little p, and then capital T, anything. Little p, capital T. There's 1, 2, 3. 3 of those. Okay? Then, what do we have? Capital P, anything, and little t. And what's this going to be? Purple dwarf. Running out of space. Capital P, anything, little t. Okay, let's see. Okay, there's 1, 2, 3. 3 of those. And then what do we have left? Little p, little t, and lo and behold, there's only 1 of those. And what is that? Green dwarf. So the ratio of our phenotypes in this dihybrid cross is a 9:3:3:1 ratio. That means 9 out of 16 are this phenotype purple tall, 3 are green tall, 3 out of 16. 3 out of 16 will be purple dwarf, and 1 out of 16 will be green dwarf. Okay, so that 9:3:3:1 ratio is 9 out of 16, because 16 is how many offspring we have. 3 out of 16, 3 out of 16, and 1 out of 16. Okay, your numbers aren't going to be 16s. So, let's see if I can give you an example real quick. I have to get out my calculator, so give me a second. So you should have 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16, okay? But maybe you have 50 plants instead of 16, so how are you going to figure this out? Nine times 50 divided by 16, you should have 28 out of 50 to give you a 9:16 ratio. You get how I did the math? Let me do 3 out of 16. 3 out of 16, and you got 50. So it's 3 times 50, 3 times 50 divided by 16. And we round off. Mine says, my calculator says 9.375. Okay, you can't have 0.375 plants. So just round off. 9. This is going to be the same because it's exactly the same calculation. Well, what about 1 out of 16? 1 times 50 divided by 16 is 3. 16 times 50 equals 3. Right? I'm almost off the paper. Sorry. Now, you may not have 50, but this is just an example of how you're going to do the calculation. If you have any trouble with the math, if you get stumped, if you're totally lost with the Punnett squares, always, 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 you can email me, m.sigmund@snhu.edu. I will answer you. We can do a WebEx call. We can talk on the phone, whatever you need. I am here for you, so just let me know. Okay, have fun with it.